Hello, back to Microbes and Biotechnology, take 2. Okay, microbiotech, microbiology. Biotechnology is actually one of the simplest and shortest units that we have in our option, and so we're lucky. The big thing to remember is that biotechnologists use a number of ways to affect cells, and these include using vectors and using retroviruses and all kinds of different things, like introducing DNA. And so it's important to keep track of everything as we go along. The first thing we'll look at is reverse transcriptase. It's basically an enzyme that you find in retroviruses. In the previous video, actually there was no previous video, so strike that out. I stated that retroviruses are a family of viruses with HIV and the feline leukemia virus as well that biotechnologists often use for whatever they want to do. This enzyme that I talk about right here is reverse transcriptase, and yeah, I'm taking this out of my notes, and I tend to write short notes, so I apologize for any problems you may be having at the moment. Retrovirus invades a host cell. So this is basically the outline. Thank you for reading. But the biggest problem in this thing, and what confused me the most, was the fact that bacteria and viruses don't have the things that are necessary to remove introns. So eukaryotes, we know through post-transcription modifications, can remove these introns as they leave the nucleus. So if you follow just this, this would actually work because you're invading the host cell. But imagine if you were introducing the genes that you wanted into the retrovirus for mass production. You don't need a PCR machine when you can have the viruses created for you. Of course, you have to make sure the viruses can't infect anything harmfully, and so you have to take some steps that I'll get to later. But the big thing is, there are two big ways that viruses are used. Two big ways. One is changing DNA for a eukaryotic cell, or mass producing genes. These are the most important ones. So yeah, you can do it through the steps I told you before. If you don't understand that, well, it's very simplified, so I can understand if it was kind of my fault. But yeah, it shouldn't be that bad. Reverse transcriptase, another diagram. Viral invasion. So this goes back to what I was telling you about how we need to control the viruses so that all of these things don't happen. Let me talk about that right now. So that is called, where am I? Yeah. That's called a viral vector. And this is the tool that they use. It's basically, they being the biotechnologists, it's basically everything that I've been talking to you about But the one thing is that viral vectors are viruses without the replication genes, and they only have the non-viral genetic material that you want. Karan's laptop is being very annoying. Vectors. That is viral vectors. So, viruses without replication genes, but with the target. I generally don't like doing these definitions because my definitions might not be the ones that IB wants. So, you're lucky I'm doing it this time. Now let's get to some of the diseases that we need to know. Cystic fibrosis is a hereditary disorder, and what happens is that there's a blockage of, and this stuff we need to know in quite a bit of detail. What happens in cystic fibrosis is that there's a defective plasma transport. So normal plasma transporters move chloride ions so that there's no sticky mucus like you can see here in the bronchi, in the lungs and the digestive system. And so you have all these problems that happen. What biotechnologists do to fix something like this is somatic cell gene therapy. What they do is they treat the individual's body cells and replace the defective ones with good ones. This is called somatic cell because you're affecting non-sexual cells, so no gametes. There is a germline gene therapy which targets gametes, but there are some ethical issues that prevent us from really getting there. It's very similar to somatic cell in its preparation and all that, but the big difference is that once you treat it, it can't show up ever again in your offspring. Somatic cell, your offspring can still have cystic fibrosis. Gene therapy. That's basically what I was telling you about somatic cell gene therapy and germline gene therapy. This picture is actually important in showing the SCID thing, so severe combined immune deficiency. And this is the gene therapy, so this is just an image of what could happen. This goes back all the way to the first things I was telling you about retroviruses and reverse transcriptase. So that's your retrovirus that goes in, deposits what's inside, which is the enzyme and the viral RNA, and then that gets absorbed into the human cell, and yeah, you create factor VIII proteins in this case. Let's go on to SCID because I really like that. You see a guy in a bubble boy. I know I'm kind of being offensive, but bubble boy is something that we'll remember him by. Bubble boy here has severe combined immune deficiency. In other words, what happened to him is, where did I have that? In other words, because of his SCID, he can't really come into contact with anything. So he has a very, very sad life. One thing that you could fix, like SCID, is through gene therapy. And gene therapy has proven to be very, very useful. And there's an 80% success rate. Let me go into details about SCID. It's caused by a blockage in ADA enzyme production. So this blockage stops you from producing lymphocytes. Lymphocytes are important in your nervous and other immune systems. It's treated through the bone marrow and umbilical cord blood. The big problem finally is ethics. Let me leave you with some questions because it's almost time for me to close. Number 1. Can good and bad uses of gene therapy be distinguished? Number 2. Who decides which traits are normal and which traits constitute a disability or disorder worthy of gene therapy treatment? At present, the high costs of gene therapy make it available to only a few people. Can costs and benefits evenly balance itself in society? Number 4, how can safety of patients be considered in the treatments that are new or experimental? So there's a 36-year-old woman who died because of experimental gene therapy, but that was a long time ago. Things have changed. And number 5, are there conflicts of interest between patient safety and the need to conduct research? So, thank you for listening. Goodbye. See y'all next time.